Okay, everybody, we're going to finish up our talk on muscle by finishing up with the two other types of muscle, which is called smooth muscle and cardiac muscle. Um, you've learned a lot about skeletal muscle, and you're going to see some similarities and differences within these two types of muscle. So let me blow this up for you a little bit. So on the panel there, where you see cardiac muscle, you'll notice that it is also a striated muscle. Okay, and remember that striations means that the muscle cell, the actin and myosin are arranged in sarcomeres. So just like skeletal muscle, excuse me, cardiac muscle is also arranged in sarcomeres with their actin and myosin. And in fact, when we study cardiac muscle in detail in the next unit in the cardiovascular system, you're going to see that much of what you learned about the sliding filament theory is similar in cardiac muscle cells, but there are a few differences. So I'm not going to go over that in this lecture and nor in this unit. We'll do that in the next unit. At the end of this lecture, though, I'll just give you a brief introduction to cardiac muscle beyond the striations here. Just some of the important structures that are important and different in cardiac muscle. The other one, smooth muscle, you can see it's labeled as unstriated, and that's because smooth muscle does not have their actin and myosin formed in the sarcomere units. Okay, so it does contract, it does have actin and myosin, but it just does that. It has a different arrangement of actin and myosin, and that's because the function of smooth muscle has to be a little bit different than what we've seen in the contraction of our other muscles. Okay, so remember, structure determines function. That's a theme of physiology. So we're going to delve into smooth muscle and exactly how it does that. So one thing to remember, if you took anatomy, you might remember from there, is that our smooth muscle tissue is arranged in circular layers around organs like the blood vessels, our bronchioles, and our lungs. Okay, so just a single circular layer like that. So when it contracts, it closes off the tube, right? In other organs like our digestive tract, ureters, uterine tubes, vas deferens, it can be arranged both in that circular layer like that, but also longitudinally as well. And so the circular layer again closes off the organ, but the longitudinal layer will shorten the organ. Okay, so that really helps to move substances through those organs. Okay, and by alternating the contractions of the layers, then that can produce those peristaltic waves of contractions. Okay, so let's zoom in on this picture, and we can see that the smooth muscle, because the actin and myosin are not arranged in a sarcomere, it doesn't have the limitations of contraction because of the sarcomere. Remember, the sarcomeres are bordered by the Z discs. It's got an M line in the middle, so myosin can only pull actin so far. Okay, but in here, the actin is 2 times more actin than myosin, and it's arranged along the length of the entire smooth muscle. And it's attached at the ends at the plasma membrane or to these cytoplasmic protein structures that we call dense bodies. You can see the little rounded structures there. Okay, the myosin, then you'll notice, gets stacked vertically, and the tails are perpendicular to the long axis of its filament. Okay, so they're just arranged a little differently, but this way they can make cross bridges along the entire length of the thin filaments. They don't have the limitation of the sarcomere. So by having cross bridges along the entire length, they can continuously pull on the actin until the cell pretty much gets crumpled up. Okay, I'll show you a picture of that in a minute. But this arrangement also allows a continuous overlap between actin and myosin, and therefore the smooth muscle can contract even if it's greatly stretched. Okay, so your urinary bladder can be stretched 2.5 times its original size. So remember, in skeletal muscle, if we stretch our skeletal muscle, that'll pull apart the overlapping actin and myosin. and you might not get any tension or enough tension, meaning enough cross bridging to create tension for the pull. But so with this arrangement, you can again stretch the smooth muscle, and it doesn't matter, you'll still get cross bridging between actin and myosin. So that's a good thing when you have to go to the bathroom and you hold it for a long time and your bladder keeps filling up and filling up because your kidneys keep producing urine and putting it in there. Your bladder then can just keep getting bigger and bigger. There's a point where you have to release it, but you can contract your bladder no matter how big it gets and releases. Okay, or like when you celebrate something with a big giant meal, your stomach can distend, can stretch really, really large, and it will still function. You have to contract in the digestive process. We'll learn more about that when we do digestion. But thanks to the arrangement of actin and myosin and smooth muscle, those organs continue to work. Okay. Here what we see is a little more detail on the actin and myosin, their arrangement. Over on the left, you see a smooth muscle cell that is in a relaxed position. Again, it's somewhat of a long cell, not longer than most of our cells, but the shortest of our muscle cells. It's tapered on the ends. That means it comes down to a point, and then that helps them arrange in sheets better and come in contact with one another better. And we'll explain why that is a little bit later. But you can also then see toward the bottom, again, that arrangement between the actin and the myosin, and you can get continuous cross-bridging and continuous pulling on the actin. And so then on the right, you see the crumpled up cell. It can really crumple up, and that's just one cell. So you can now imagine with the circular or longitudinal arrangement, you can really either close down an organ or shorten it with this type of contraction. So while there's continuous cross-bridging, there's still control over it. So we're going to look at the mechanism of how we control the cross-bridging and the power strokes to occur. Okay. So first, let's remind ourselves of the different types of filaments, and most of them are the same from skeletal muscle. So you still have your myosin filaments that are bundled together, and these are referred to as the thick filaments. You still have actin filaments, and these are thin. However, there's no troponin. So troponin is not the molecule that's controlling the cross-bridging here. Okay, so no troponin in smooth muscle. Okay. And then the intermediate filaments are those that I mentioned that help form the cytoskeletal framework and supporting the shape within there. They're not directly participating in the contraction, so we're not really going to talk about those in the contraction cycle. So here's what it looks like inside of a cell with the contraction. So let me walk you through, and you're going to see some new molecules that are here. Okay, so this would be like if we looked inside of a smooth muscle cell. Notice the things that are the same. You see the myosin that are in blue, the actin is in green, but they're arranged a little differently. We have a sarcoplasmic reticulum, the SR, but the difference now is they're very much reduced in the smooth muscle cell. They're very small. So it still contains calcium, but it doesn't contain enough calcium. The calcium primarily is coming from the interstitial fluid, right? So that's extracellular calcium. So there is a voltage-gated calcium channel there. So when we'll still have nerves stimulating the cell membrane of the muscle cell, and that's going to open up these calcium channels and let the majority of calcium in. So we still get calcium from the SR, but they're not big enough. So we get most of our calcium from the extracellular fluid. Okay, now calcium, I mentioned already that we don't use troponin in this process. So calcium binds to a different molecule called calmodulin. So calmodulin occurs in the cytoplasm, and it isn't activated until it binds with calcium. So once calcium gets released into the cytoplasm, it binds with calmodulin, and now it's active, and it can activate an enzyme called myosin light chain kinase, MLCK, you see in the middle there. And myosin light chain kinase, its job is to phosphorylate the myosin. So it's going to hydrolyze an ATP to phosphorylate the myosin. And it's going to do that on a specific area of the myosin called the myosin light chain. So let me show you the next slide, which will have that detail in there. It shows the same steps, but in a flowchart format, which is what you see here. And so let me skip down to the bottom to show you the myosin light chain, and then we'll go through each of the steps again. So here you see your myosin looks like that, shaped like a club again. It's got a head, but in the neck region it has what's called myosin light chain. Okay, these are actual chains of proteins that are attached to the heads of myosin molecules near that neck region, and they're light in weight. So that's why they're called myosin light chains. Okay, they are found in skeletal muscle, but they're not very important. But in smooth muscle, this is what regulates the cross-bridge cycling. So in order to do that, they must be phosphorylated, meaning they have to have that inorganic phosphate bound to it in order for the cross-bridge to occur. And that phosphorylation occurs when ATP is hydrolyzed. So remember, that means an ATP, the inorganic phosphate, is removed off of an ATP, and then it will be attached to the myosin light chain. Okay. Notice at the head, you see the inorganic phosphate attached to the myosin light chain, but at the end of the head, there's a different ATP that produces the power stroke. Okay, so the phosphorylation of the myosin light chain permits the binding, permits the cross-bridging, but then it's the ATP that hydrolyzes on the myosin head that produces the power stroke. Okay, so again, in order to phosphorylate the ATP to connect it to the myosin light chain, we need the enzyme myosin light chain kinase. Okay, and that is inactive until calcium binds to calmodulin. So I kind of went through that flowchart backwards. Let's do it forward. So again, calcium enters the cytoplasm from the extracellular fluid and the SR. Most of it's coming from the extracellular fluid. It binds to calmodulin that's already in the cell. That combination then activates the myosin light chain kinase, an enzyme. That enzyme now hydrolyzes the ATP and phosphorylates, meaning it takes the inorganic phosphate and connects it to the myosin light chain. So now the myosin light chain is phosphorylated. And now the cross-bridging can occur. So that means it can lift up the head and make that cross-bridge with actin. It also needs the ATP on the head and hydrolyze there so that the power stroke can occur. Okay? If that is a better way for you to look at it and study, or if you like this picture, again, it's the same thing. You can see calcium coming from the interstitial fluid, or ECF, and the SR. It's binding to the calmodulin. Calmodulin is activating myosin light chain kinase. That's phosphorylating the myosin light chain. And another ATP also then binds to the head so that the cross-bridge, the power stroke can occur. Okay, so the phosphorylation of the myosin light chain is allowing for the cross-bridge to occur. The ATP at the end of the myosin head that hydrolyzes is causing the power stroke. And that's all I want you to know for this one. We went into a little more detail about the relaxation of skeletal muscle. This is as far as I want you to go for smooth muscle. And so sometimes it helps then to make a comparison of the two. So when you compare the two, again, you do need to know the process of both skeletal muscle and smooth muscle. So one of the differences is that well, what's the same is that both muscle types need to be excited. Okay, so they need to get um, excited by a neuron. And we're going to look at that in just a minute on smooth muscle. Okay, what's also important for both is that calcium needs to come in there to start this process. However, where calcium comes from is different. So in smooth muscle, calcium comes from mainly the extracellular fluid. In skeletal muscle, it all comes from the SR. Okay? Also, then the next steps that occur are different in smooth muscle and skeletal muscle. In smooth muscle, it's a series of biochemical events, okay, activating the enzyme and phosphorylating myosin. In skeletal muscle, it's a series of physical events that occur. It's the physical repositioning of troponin and tropomyosin. And then we can get our cross bridging and power strokes. Okay, ATP important in both. Okay, phosphorylation of myosin at the cross bridges of the thick filament. Okay, in smooth muscle. So you can see a lot of similarities and differences there, but so you really need to understand just the process of both. I'm not going to ask you what's the same and what's different as smooth muscle and skeletal muscle. If you learn it more as a process of each one, that might be easier for you to do. Alrighty. So now let's look at smooth muscle, the tissue as a whole, and we're going to see that there's actually two major types of smooth muscle. Okay, it's called multi-unit smooth muscle or single-unit smooth muscle. So multi-unit smooth muscle consists of multiple discrete units of muscle cells that function independently of one another. Okay, so they can be stimulated separately by nerves. That's almost similar to the skeletal muscle. Okay, so again, what we're doing is we're stimulating different muscle groups and different areas of that organ, of the smooth muscle portion of the organ. Okay, in single unit, and it's sometimes called visceral smooth muscle, when it gets excited, all of the cells contract as a single unit. Okay, they all contract at once. When several cells act as one, as a single unit, we call that a syncytium. Okay, this should be in your book, and some of the anatomy professors I know talk about it. It's spelled SYNCYTIUM, syncytium. It just means all the cells are acting as a single unit. We're going to see the heart does the same thing too. So let me explain a little bit more in detail on both multi-unit smooth muscle and single unit smooth muscle. Okay, so just like skeletal muscle, it's neurogenic, means it has to be stimulated by nerves. Okay, but these nerves are laid out in a way that, again, they can form discrete units that function independently of one another. Similar to the motor units that we saw in skeletal muscle. We don't call them motor units, though, in the smooth muscle. Okay, so they're separately stimulated by nerves to contract, and that it happens with the autonomic nervous system. The autonomic nervous system are the nerves that control multi-unit smooth muscles. Okay, and here you can see a picture of the nerves, and you can see that they're structurally different than the motor nerves that we saw with the skeletal muscle. So instead of the synapses, that synaptic bulb at the end, these form what are called varicosities, but it's functionally similar to those synapses that we have seen. Okay, again, they're just anatomically different. So functionally, they're the same because that's where the vesicles contain that produces the neurotransmitter, and the vesicles merge with the cell membrane, and it releases it into a synaptic cleft, which then attaches to receptors on the smooth muscle cell membrane. Okay, so again, very similar function, but just the anatomically different, anatomically in the form of these varicosities. Okay, and multi-unit smooth muscle tends to be found in organs like the walls of your large blood vessels, in the airway to your lungs. I mentioned the bronchioles. In the muscle of the eye that adjusts the lens for near or far vision. The iris of your eye, what else? The base of the hair follicles, right? So think of the function of these areas. These are areas where you might want to contract some cells, but not all the cells, right? So you might want to contract large blood vessels are can span large lengths of your body, and you might want to contract one area but not the other. Okay. Same with the muscle that adjusts the lens for near or far. You might want just a small adjustment or a big adjustment. So it kind of gives you a different, it's not strength of contraction now, but different areas of contraction. So you can make fine adjustments. Okay, maybe that's what's important here. You can make fine adjustments. Okay, the next slide shows single unit smooth muscle at the top, but I added the multi-unit smooth muscle at the bottom so you can compare. Okay, so again, these are also stimulated by nerves, but the nerves are embedded within the organ. Okay, so it's not controlled by the ANS. And we'll see this when we do the digestive system. If you remember, there was a part on that flow chart for the parts of the nervous system. There was the enteric nervous system. So that's really where this one's coming in. We'll get more into that when we do the digestive system. Okay, so it's self-excitable. That's what it means. It doesn't need to come from a nerve from the ANS. This nerve tissue is embedded within the smooth muscle. And they still form varicosities instead of synapses that we're familiar with. So because of this arrangement, the fibers become excited and then they end up contracting as a single unit. And what helps with that then is that the smooth muscles are connected by gap junctions. And you can see those lines are pointing to some of the gap junctions. Okay, so again, that's a space created so that the cell membranes of two adjacent cells open up and the cytoplasm is continuous. So you get a faster flow of this excitability of the ions moving from one cell to another to excite the cells much more quickly. That's another way that they function as a syncytium. Okay, this is a much more energy efficient way for the cells to contract, and this is well suited for forming the walls of, again, these large distendable organs, hollow organs like your digestive, like your stomach, and things like that. You want that to contract as one unit. Okay, so you can contrast that with the multi-unit smooth muscle at the bottom. I like that picture because it shows the nerves traveling in between smooth muscle cells, and so it shows that separation of some of the smooth muscle cells so that they act independently. They can act independently of other muscle cells. Whereas in the single unit smooth muscle cell, they're acting as one single unit. They'll contract together. In the multi-unit, they can contract in multiple different ways. Okay, so that's it for our smooth muscle. I'll just end this with a little bit on cardiac muscle. Again, I'm not going to get into depth of cardiac muscle until we do the cardiovascular system next. But for now, what you should know for this unit is that cardiac muscle is the only type of muscle found in the walls of the heart. Again, it is striated. Striation means there are sarcomeres, okay? Striations are the stripes that you see there, and that is formed by the sarcomeres. So if it has striations, then it has sarcomeres. Okay, don't forget that. Okay, cardiac muscle cells are also interconnected by gap junctions. And so again, this is going to allow the cells to function as a unit. Okay, you want your heart cells to contract as a unit. Otherwise, you'll get fibrillation, right? You want your heart to contract all at once so that blood gets pushed out efficiently. Okay. The other thing that's different, we do have elongated muscle fibers, but they're branched. Instead of being one long cylinder in a skeletal muscle, these are branched. Okay, and again, that just helps form the structure of your heart. The cells then can wrap around and form that structure of your heart. Okay. Also notice the intercalated discs in between the cells, intercalated discs, and that helps to bind those cardiac muscle cells together because they're constantly contracting, relaxing, contracting, relaxing. That's the heartbeat, right? And you don't want those torn apart. So that helps to bind them together really closely. Okay, and the cardiac muscle cells, they are innervated by the autonomic nervous division. So while the heart muscle cells themselves can contract, they don't need the outside stimulation for the contraction. We'll learn how they do that in the next unit. But the autonomic division can speed up the contraction or it can slow it down. So it just modifies the electrical activity in the heart. Alrighty? And that's it for muscle. That's it for this unit. Happy studying, everybody. I know you can do it.